Hormone supplements don't always deliver. Here's why. Many individuals with low hormones, whether from aging or another medical reason, try supplemental testosterone, estrogen, or progesterone. The hope is that more hormone should restore sleep, energy, and mood. But results are often mixed. Some notice improvement, others see little change, or even new sleep difficulties. Part of the reason is that all three hormones affect sleep, and affect sleep in different ways. Testosterone can support deep sleep under physiologic replacement, while supraphysiologic dosing can disrupt sleep architecture. Estrogen modulates circadian timing, body temperature regulation, and elements of REM sleep patterns. Progesterone enhances calming brain activity through its metabolites' interactions with GABA receptors, helping reduce wake after sleep onset. These roles are generally well characterized, yet boosting hormone levels alone doesn't always translate into better nights. One of the core pieces often overlooked is that hormones don't act directly. They must first be received and processed by the body's tissues. When it comes to sleep, this binding and signaling take place in brain regions such as the hypothalamus, basal forebrain, and brainstem nuclei, areas that regulate circadian timing, arousal, and the depth of sleep. Hormone function relies on utilization, not just levels or production. You may recall, I've mentioned in a preview article that hormone function is not determined by hormone levels or hormone production alone. Once a hormone is made by the body, it still has to move through several stages, being carried through the body, converted into usable forms, delivered to tissues, used by the tissues, and eventually removed from the body. All of these steps matter, but here we'll focus on receptor sensitivity and utilization, the ability of target tissues to recognize and respond to the hormone once it arrives. In simplified terms, receptors are the entry point for hormones to act on a cell. In more specific terms, a receptor is a protein on or inside a cell that recognizes a hormone molecule and enables the hormonal signal to be transmitted. There are three features of hormone receptors that affect how effectively a hormone signal is transmitted once the hormone reaches a cell. Receptor number. Expression: how many receptors are present in a tissue, such as the hypothalamus or hippocampus. Fewer receptors mean less capacity for hormone action, regardless of circulating levels. Binding affinity and activation efficiency. How strongly the hormone attaches and whether the receptor activates properly once bound. Aging and oxidative stress can alter expression and impair signaling efficiency. Downstream signaling capacity. The ability of the receptor hormone complex to turn on genes, produce proteins, and adjust neural or metabolic functions. If this cascade is weak, the hormone's impact remains limited. This is why hormone supplementation, whether testosterone, estrogen, or progesterone, often produces variable results. Supplementation changes circulating levels, but does not address receptor sensitivity. If receptors are sparse, unresponsive, or poorly coupled to downstream signaling, additional hormone may not improve sleep or other changes in the way one expects. And that leads us to the question of what shapes receptor sensitivity over a lifetime. SSO, what affects hormone receptor sensitivity? Several factors affect how responsive hormone receptors are, and why two individuals with similar hormone levels can potentially have different sleep outcomes. Genetics set the baseline. Variants in receptor genes shape sensitivity from birth. For example, the androgen receptor gene has regions called CAG repeats. A shorter repeat length generally produces more responsive receptors, while longer repeats reduce sensitivity. Genetic variation in estrogen and progesterone receptors also influences signaling, though patterns differ from androgen receptors. This creates individual differences in how responsive someone is to a given hormone's effects, on sleep or otherwise. Aging reduces receptor sensitivity. With advancing age, receptor expression in brain tissue declines. This means there are fewer receptors available for hormones to bind. Human basal forebrain studies show nuclear androgen receptor staining declines with age. Estrogen and progesterone receptor expression also change with age, accompanied by alterations in binding efficiency and downstream signaling. Why this matters for sleep. The receptors for testosterone, estrogen, and progesterone are located in brain regions that impact circadian timing, stress recovery, and sleep depth, especially the hypothalamus and hippocampus. As receptor number and sensitivity decline, the same circulating hormone level produces a weaker biological effect. Declining receptor sensitivity may be one of many contributing factors to why sleep becomes lighter and more fragmented with age, especially when hormone levels are normal. So, is there anything we can do to maintain hormone responsiveness? The good news: hormone receptor sensitivity can change. What research shows the encouraging news is that receptor sensitivity is not entirely fixed. While genetics set the baseline and aging reduces expression, there is some evidence that activity and environment can shape how responsive receptors remain. Animal studies. In rodents, estrogens promote synapse and spine formation in the hippocampus, and enriched environments and neural stimulation can modulate the magnitude of these estrogen-linked plasticity effects. Androgens acting through their receptors also support hippocampal structure and plasticity, such as maintaining dendritic spines, and studies show that mild exercise elevates local dihydrotestosterone, DHT, and requires AR activity for exercise-induced neurogenesis. These models illustrate that receptor responsiveness is not static. Even in aging tissue, environmental inputs can enhance or preserve receptor-driven plasticity. Human studies. In muscle, resistance training often increases AR activity, and in several studies increases AR content. Individuals with higher receptor expression in muscle show greater gains from training, demonstrating that receptors can be upregulated and functionally amplified in peripheral tissues. The caveat: direct human evidence for increasing receptor number or sensitivity in the brain is limited. Most data come from animal studies or from peripheral tissues such as muscle. Still, taken together, these findings suggest that receptors are dynamic. While the evidence for brain receptor plasticity in humans remains indirect, lifestyle factors that influence receptor function in muscle and in animal brain models make it biologically plausible that similar mechanisms could support human brain signaling. In short, receptors decline with age, but they are not beyond influence. What we do daily can either diminish their responsiveness more quickly or help preserve and even support their function over time. Actionable steps for supporting hormone receptor sensitivity. If receptor sensitivity influences how well hormones shape sleep, the next question is, what can be done? While direct human evidence on modulating receptor function in the brain is limited, several systemic levers have support. Resistance training in human muscle. Resistance training increases androgen receptor expression and strengthens signaling efficiency. Regular, progressive sessions that challenge large muscle groups are associated with favorable receptor adaptations in muscle. Reduce oxidative stress. High oxidative stress can disrupt receptor expression and signaling by damaging proteins and impairing coactivators. Approaches that lower oxidative load, such as aerobic conditioning and limiting excess alcohol, create a more supportive environment for receptor function. Support synaptic plasticity. Hormone receptors operate within synapses, where they interact with neurotransmitter systems to shape circadian rhythm and sleep depth. Animal studies show that both estrogen and androgens influence synapse formation in hippocampus, and that physical or cognitive activity helps maintain synaptic plasticity. While human brain data are sparse, it is plausible that preserving synaptic health indirectly supports receptor sensitivity in sleep-related circuits. These strategies may not change receptor expression in the brain overnight, but they influence the broader conditions that affect how effectively hormones can act. Hormone receptors are the gatekeepers of hormone effects on sleep and beyond. Hormone production and the circulating levels it creates is necessary for optimal hormone function, but it is only the starting point. Whether testosterone or estrogen improve sleep depends on receptor sensitivity, the number of receptors available, how efficiently they bind, and how well the downstream signal is carried forward. Direct human data on changing receptor sensitivity in the brain are still limited. Yet the broader picture is clear. Lifestyle choices such as resistance training, reducing oxidative stress, and maintaining synaptic plasticity shape the environment in which receptors operate. In this way, you can improve how effectively your existing hormones influence sleep without needing higher doses. Bottom line, production sets the supply, sensitivity determines the impact. By supporting receptor function, you improve overall hormone function. That means the hormones you already have, or those you supplement, can more effectively support sleep, energy, overall vitality, so you feel your best for decades to come. If this episode was helpful, consider liking and subscribing so you'll be notified when new ones go live. And if you'd like to help more people move past 3 a.m. wake-ups, share this episode with someone who's been struggling to stay asleep. To learn more about Sleep OS, my flagship system built to help you sleep through the night, even when good sleep hygiene hasn't worked, visit thelongevityvault.com. The link's in the show notes.